you very much for inviting me and for giving me the opportunity here to present the European perspective on qualification. And maybe just a small correction. I'm chair of the 3Rs working party at the European Medicines Agency, not a non-clinical working party. So I'm gonna try to advance them. Okay. Okay, so the usual disclaimer that I'm speaking on my behalf. And just the overview of the presentation. So I'll be setting the scene a bit of the position of Europe and the towards 3Rs and the general framework for pharmaceuticals, EMA and the 3Rs. I'll give you some insights on our perspectives on regulatory acceptance and then on regulatory acceptance of complex in vitro models, and I'll finalize with some take-home messages. So just in a nutshell, we have a directive which is currently under revision that gives the community code related to medicinal products for human use in Europe. And the reform that's ongoing is really a large-scale reform of the EU pharmaceutical legislation. And there a directive and a regulation will ensue that will enshrine really the principle of the 3Rs, replacement, reduction, and refinement of animal experiments. And this all refers to the Directive 2010/63 of the European Parliament and the Council on the protection of animals that are used for scientific purposes. And this directive clearly states that in Europe, when an alternative is available and is at least qualified or validated, it has to be used instead of an experiment on a live animal. So just a recap, probably I'm kind of repeating what you already know, that the assessment of medicinal products is a benefit risk assessment based on quality, non-clinical and clinical data, and in Europe supports clinical trial applications, marketing authorization applications, and of course clinical use later on. And our non-clinical program is in line with phase of clinical development, duration of treatment, and the type of modality or indication. And on the right-hand side, you see clearly the drug development program. And when we're discussing context of use, that's exactly as what we are referring to is where is this tool used in the drug development program and what kind of decisions are being taken on the basis of the data that result from this tool. So just a short word on the position of the European Medicines Agency towards 3Rs. It's not a new thing. We had already a joint expert group on 3Rs set up in 2010. And as you can see, this culminated in 2022 in the creation of a standing 3Rs working party, following also the statements in the EMA regulatory science strategy. And we have now a new work plan under development for the next years. And if you scan the QR code, you can find all the details on our website. So what are the tasks of this 3Rs working party? We want to really promote regulatory integration of all tools that are 3Rs compliant. So this covers quite a lot. Not only in vitro tools, but also in silico tools and reduction and refinement proposals. And in fact, the topic also of today, we want to promote and support the qualification of novel approach methodologies, which means not only qualify the tools once they're there, but also aid in the design of qualification criteria to support later regulatory acceptance. And we feel that it's really important to do this through stakeholder interactions. So we also want to foster these and also want to foster international collaboration. We have different tools to interact between stakeholders, and I'll come back to that later on in the presentation. And I just want to draw some attention to the training that we want to set up, because that's really a point where we feel it's important to emphasize, since regulators need to be trained to be able to assess these novel tools. And also we want to do this in collaboration with our academics, and therefore we set up a special expert community to this end. So what is regulatory acceptance for us? We have a guideline on principles of regulatory acceptance of 3R's testing approaches that clearly defines regulatory acceptance in two ways. The first one is the incorporation of a novel approach method in a regulatory testing guideline, and this is a bit the golden grail, in fact. It's the incorporation either in an ICH, a European Medicines Agency guideline, a veterinary ICH guideline, or in a monograph of the European Pharmacopoeia. But there are other ways to accept data coming from a novel tool, and the second way is to accept the data for regulatory decision making for a specific drug development program. And this is rather on a case-by-case basis. This can be to support a clinical trial application or later on to support an actual marketing authorization application. And often these requests are submitted to us through targeted product-specific scientific advice. Now, what are the criteria for regulatory acceptance? And these are also found in this guideline. You have to have a test methodology that is clearly defined with a protocol and endpoints. And then, of course, the word of today, you need to define the context of use of the method. So really describe the circumstance under which the testing approach will be applied in the assessment of either a human medicine or a veterinary medicinal product. And this also implies to describe the limitations, because as any tool, this novel tool will also have its limitations. So once the context of use is clear, the relevance within that context of use needs to be described, including the accuracy and the reliability and the robustness of the method needs to be determined. Now, since we have this context of use so prominent, the regulatory acceptance criteria, of course, are driven by that particular context of use. And when we're thinking about the regulatory use of a novel tool, it can be quite diverse. And as we see evolution that is ongoing, the integration of these novel tools or the data from these novel tools in weight of evidence approaches, as are already described in some ICH guidelines, is probably the most evident route for progress. On the other hand, you have the integration in integrated testing strategies, such as described for the assessment of cardiovascular safety pharmacology endpoints, and only on a rare, in a rare case, there will be one-to-one replacement. So what are these contexts of use? And there are many. So what we try to do from our side is to provide an overview of all the current regulatory testing requirements for medicinal products for human use. We have a similar one for veterinary use, and identify the opportunities for implementation of the 3Rs. And as you can see, it's a kind of tabulated overview, and I've lifted out the safety pharmacology guidelines, and there it refers to the ICH S7BE14 question and answers that was very recently published, and that defines a context of use for NEM data as part of an integrated strategy. So building on this context of use, best practices and principles can be identified that can serve as a basis for qualification of tools that are used for this particular purpose. Now, in Europe, there was also a large-scale project, the ORCID project, that had the intention to build a roadmap for organ-on-chip technologies in Europe. And as a part of this exercise, there were workshops that were around the identification of potential contexts of use for organ-on-chip technologies. And this could be extrapolated to complex in vitro models. And what you can see is that from these multi-stakeholder workshop considerations, different contexts of use were already identified, going from the identification of disease mechanisms to drug efficacy and toxicity, up to personalized medicine. So all these contexts of use will entail different approaches to qualification criteria design. So coming from the consideration on the regulatory acceptance to the different entry ports to engage with the European Medicines Agency with regards to NAM acceptance, such as complex in vitro models. So on the first hand, you have an early interaction that is possible through the EMA Innovation Task Force on 3Rs. This is an entry port that focuses on the encouragement of the development and the regulatory acceptance of these novel tools. And it really provides a forum for early dialogue between multiple types of stakeholders and the regulatory network and the EMA. The outcome of these meetings is really informal, non-legally binding guidance that will give you insights on the need for a qualification package, and if you need one, the design or further elaboration of one. So these meetings are free of charge and very open to any type of application. So in the context of a horizon scanning that was done at the level of the EMA Innovation Task Force, we reviewed the briefing meeting requests that were submitted to the ITF between 2019 and 23, and which contained 3Rs related topics. And as you can see on the left-hand side, the amount of requests has clearly increased from 2019 up to 23. And when we kind of dissect what kind of R is covered in these requests, you can see that most of the time this concerns replacement methods. And within these replacement methods, we can see that the majority of tools that are discussed are in vitro tools, followed closely by the combination of in vitro and in silico tools. So the next step is, of course, the actual qualification of NAMs, and this is governed by the guideline on the qualification of novel methodologies for drug development. And this always starts, of course, by a submission of a qualification request to the EMA. And then a multidisciplinary qualification team is composed that is composed of experts drawn from the European network, and it takes into account the particularities of the method at hand. Now, if it concerns a 3Rs tool, of course, 3Rs working party and 3Rs experts from the regulatory network will be involved. And we can also involve other international agencies such as the FDA, for instance. So what's the outcome of a qualification? And there are different possibilities. First of all, you can have a qualification advice that will give you insights on future protocols or methods for future method development towards qualification. So that means that the package is still a bit premature and that further work needs to be done. Now, we can have this advice as a confidential advice, but we can also, on the basis of the advice, redact a letter of support that can be published on the EMA website, and that will say that, okay, the method is not yet at a maturity of being qualified, but it is promising enough to be further pursued. And the aim of these letters of support is to really encourage further data sharing and building further data collection so that the package is brought to a higher level of maturity and can end up in a qualification opinion. And that's really a full-scale review of the data that ends with an opinion that is then published on the EMA website and is a formal statement that the novel tool can be used under a predefined context of use. So this, of course, leads to the question, what is a mature qualification package? Because the level of maturity will hence determine whether we end up in an advice or we can proceed immediately to an opinion. And then finally, at the level of a product-specific NAM use, we can, or a pharmaceutical company in combination eventually with a method developer can come for scientific advice. And therein it will be the consideration for a 3Rs implementation in a specific drug development program. This also includes deviations of possible existing guidance. guidelines. And the relevant domains that are targeted for 3Rs are, of course, the quality and the non-clinical testing programs. And scientific advice can be requested at any time during drug development, starting from very early pre-phase 1 up to phase 3 or guidance for later marketing authorization application. So clearly, pharmaceutical industry, academic groups, SMEs, and so on can apply for these types of advices. And here again, there is a possibility to have parallel EMA and FDA advices. So we have different possibilities to interact, and I only would like to touch a word upon the possibilities for further data collection. So in our guideline on the principles of regulatory acceptance, we have included a process called the voluntary submission of data, or the so-called understood safe harbor process. And this means that specifically for 3Rs methods, such as a complex in vitro method, there is a process where data that are obtained with the complex in vitro model can be generated in parallel with the data that are generated using the conventional per guideline methods. And the outcome, so the data that are generated, will not be used for product-related regulatory decision-making, but will be used specifically for the purpose of evaluating the NAM for possible future regulatory acceptance. And all the data that are generated can be submitted for review and then decision-making on a possible regulatory acceptance status or a qualification. Now, this is really a process for the evidence building in support of the qualification, or later on for further fine-tuning of the actual use or the context of use of the qualified NAM. And it's clear that the conditions to set up such a process should be discussed on a case-by-case basis during either an innovation task force meeting or during a qualification of novel methods process. So how do we progress towards regulatory acceptance, and what does the EMA 3Rs Working Party want to do to facilitate this? So we've devised a kind of multi-step process that starts from multi-stakeholder interactions over context of use-based qualification up to regulatory acceptance. And let's first target the multi-stakeholder interaction. So in the context of complex in vitro models, we have organized multi-stakeholder workshops that are focused on defining the requirements for regulatory acceptance of these complex in vitro models. And we have organized 2 workshops over the years. The first was very early in 2017, and the last one was in the beginning of this year under the Belgian presidency of the Council of Europe. And if we pull all the take-home messages from these 2 workshops together, we can see in general there was really the request to have specific qualification guidance and endpoint-specific performance standards and definition of reference compounds in line with context of use. Now, the way how to progress was really the message: start simple and progress stepwise. And please ensure that there is the necessary flexibility in your qualification criteria for continuous applicability of those, because we know that the technology and the science is progressing continuously. So if we make qualification criteria that are too rigid, we will not be able to follow up with science. And also data sharing was put forward as a very critical tool to generate further evidence and to substantiate the data building for qualification. And finally, of course, international harmonization is key, and I think it's really great that we can discuss this here together today. So on the more specific side, with regards to context of use qualification, it's clear that context of use qualification should not be superimposed on biological and technological characterization, but that we need really this basic characterization to be able to progress to further context of use qualification. And also we need these tailored criteria to address the specific needs. So how do we go about to translate what we see in the models to the clinical situation, which is what we want to predict, of course. So it's clear that the context of use will drive the definition of what is a gold standard and the reference compounds, and that in this aspect we need to see what is the applicability of the clinical human biomarkers, and if we can't rely on those, whether we can rely on mechanism-based translation. Moreover, there needs to be a careful selection of reference compounds, and these need to be lists, preferably that are quite harmonized and approved and well curated, and that include a reflection on the different types of therapeutic modalities which we are confronted with, all for which the models will be used. And finally, there is, of course, a key role for in vitro, in vivo modeling in relation to predicting human exposures and relating the data from in vitro to the human situation. So with regards to stakeholder interaction, we cannot forget the worldwide aspect and the global harmonization aspect. In that context, 3R's working party at the EMA took the initiative to set up an international medicines regulator working group on 3Rs. And we had a kickoff meeting in January of this year. And as we speak, the terms of reference have been drafted and are currently under approval in the different agencies, and which you can see on the lower side of this slide. And if we take out the basic message, of this group is really to foster a consistent global approach across regulatory jurisdictions to achieve internationally harmonized 3Rs recommendations and assist in the implementation of new alternative methods for testing of human and vet medicinal products wherever possible. So going now to the context of use-based qualification, and this of course starts with the drafting of regulatory acceptance criteria. And in this context, EMA has drafted a concept paper to revise its guideline on the principles of regulatory acceptance of 3Rs testing approaches in order to define really the critical 3Rs related terminologies, because in the discussions we have had, we saw that there is a lot of confusion, I would not say, but at least different interpretations on what is qualification, what's context of use, what's the difference with validation, and so on and so on. So we will try to kind of make a basic list of terminologies that, at least from our side, it's clear what is meant by what. Further on, we will include annexes that provide regulatory acceptance criteria for complex in vitro models for specific contexts of use to be applied in the pharmaceutical area. And the intention is really to start, first of all, with the two contexts of use, predicting drug-induced liver injury and predicting safety pharmacology, cardiovascular safety pharmacology endpoints. So moving on to the early dialogue via the Innovation Task Force. And again, from this horizon scanning that was carried out, we looked at the type of in vitro tools that were submitted and which organ models were mimicked. And you can see, as expected, most of the tools were liver models, followed closely by brain, heart, musculoskeletal, and other types of models. And if you see on the right-hand side, you can see that most of the tools were 2D in vitro models, but they were almost equal amount of organ-on-chip technologies submitted. And I think if we draw the numbers again for 2024, we will see that the number of applications for organ-on-chip technologies has significantly increased. Finally, coming to the EMA qualification of NAMs, and from our side, we have had no qualifications yet for complex in vitro models, but looking back also at the experience that we had on drafting qualification criteria, and I'm referring here to the ICHS 5 guideline, I think there are a number of lessons that we can learn from past experiences, because also for this S5 guideline, where we have really clear qualification criteria, we can see that we have no qualified methods yet. So what could this be due to? We have probably a lack of clarity in the qualification requirements themselves. There is uncertainty with regards to global harmonization and global acceptance. There are considerations with regards to limitations of the different tools that can be qualified, and there is a general reluctance to share the data. And then finally, there might be perception issues, that things that are really in the guideline and written and expected by the regulators might be perceived as differently by end users or industry partners. So this only gives us the conclusion that we really need to communicate clearly upfront, consult the stakeholders at initiation of the guideline drafting, and promote really the sharing of the data in parallel with in vivo studies, so that we can build evidence as we go. And we really need to increase the very limited experience that regulators currently have, especially with these novel tools, to build trust also with regards to acceptance. So to wrap it up, context of use, and that's what we will be discussing today. So it's really the key and the starting point for further work. And then finally, also think about integration of the data in weight of evidence approaches. They will not give all the answers to make a regulatory decision, so they will have to be considered in relation to other data that might come from other types of studies, that might come from literature, and so on. So also the early interaction with the regulators is important, and the submission or sharing of the data to the authorities is really encouraged. And we need to have a proactive approach, so reflect on the criteria for these complex in vitro models, exactly what the intention of these types of workshops are. Engagement and open dialogue with stakeholders is key, and let's learn from the past and avoiding making the same mistakes. And with that, I thank you for your attention.